Good evening, everybody, and welcome to Cafe Scientifique, Silicon Valley at SRI. I'm going to let you know we're videotaping tonight's event, and we will post the video to our YouTube channel, so where you can also find lots of videos from our past events. So if you want to check us out later, pick up a blue bookmark on the way out. It has our YouTube address along with our other social media URLs. And I'd like to take this opportunity to tell you about our next Cafe Sci after this, and then we'll introduce tonight's speaker. On September 10th, Dr. Michael Snyder of Stanford will discuss personalized medicine. Dr. Snyder sequenced his own genome to examine the links between his DNA and the biochemical fluctuations in his body over time. It changed his life. His genome sequence revealed his risk for type 2 diabetes. When he was later diagnosed with the disorder, he was ready. Dr. Snyder will discuss the big questions in personalized medicine. Should people get their genome sequenced? And how could that help their health? Join us on September 10th for an enlightening and thought-provoking presentation. Okay, so let me tell you about tonight's speaker who will engage us on another important health topic, clinical trials. Dr. Manish Kothari has worked on all aspects of clinical trials over the past 20 years. He has worked at several sites as a clinical trial sponsor. He founded an innovative telemedicine company for clinical trials and designs and builds medical implants. He joined SRI last year as our program director for robotics and health to commercialize SRI medical technology products. Dr. Kothari received his Ph.D. from Cornell University and conducted postdoctoral studies at UC San Francisco. So please help me welcome Dr. Kothari to the podium.
Thanks, everybody, for coming today. Hope I present a lively presentation, taking you on a romp through history, going all the way back to when clinical trials started, and all the way in the future, where what they will like to be. So, just a quick set of disclosures. I'm an employee as mentioned at SRI. I have been a regular consultant to a number of large, pharmaceutical and medical device companies. I own stock in a few companies, and I'm an advisor on a few as well. There's some inspiration and credits I would like to give to Dr. Atul Butte from Stanford, Dr. Chas Bujra from Oxford, Roger Lewis and Dr. Berry, some of whose slides are being presented here. The bottom just shows some of the products I have been involved with, all the way from spine implants to some medical imaging techniques and innovative clinical trials at your home where we're studying overactive bladder and you don't have to come to the hospital. So let's start. Clinical trials started a long, long time ago. The first one is actually in Babylon, when King, and let me get the name right, Nebuchadnezzar, was talking with Daniel, and he said, the right food for kids is meat and wine. Daniel said, I think meat and pulses and water are better. So they decided to have a trial. 3 days later, after a breakfast of meat and wine, the kids were not doing too well. The other kids that Daniel spoke to were doing great. The trial was decided, and they switched the diet to meat and pulses. So that's the first trial. That's the first recorded trial. To pulses and water instead of meat and wine. Sorry? Legumes. There you go. Fast forward 17 centuries, and now you go into England, UK. Scurvy was a big problem. You know, we have all these beautiful pictures of romantic sailing ships going across the water. We even have Russell Crowe out there looking great, dashing with beautiful hair. But the reality was scurvy was pretty nasty. Your teeth fused together. You had other things going wrong in all your cartilaginous parts. And nobody knew why. Everybody was wondering what to do. And the daily allowance of rum or gin was not making a difference. Dr. James Lind was the first person who noticed this and decided to run a clinical trial. It was the first controlled clinical trial that is recorded in history. He had 6 different groups. He He fed some of them cider, some of them garlic, some of them seawater, some of them vinegar, some of them hard oranges and lemons, and some of them hard cider. Now let's say this, the people who were happiest were the ones who got hard cider. The people who did the best in the long run were oranges and lemons. We all know this story. What we also know is that he couldn't convince the British Navy to give him oranges to lime juice for 50 years because it was too expensive. So it was only after 40 to 50 years that they finally recognized this, started giving him lemons, and we all know that's where limeys came from, the name, and they switched to limes because it was cheaper than lemons. I'm going to fast forward a little bit here. Let's fast forward a couple of hundred years and talk about Chicago, because that's the next step in the history of the clinical world. Chicago was the center of the railway in the late 19th centuries in the US. And it was around that time that refrigerated railway lines were also created. What happens there is you have lots of prairies, increased demand for meat, you have refrigerated lines, and you have the railroad. Perfect environment for the Union Stockyard to come about, and lots of cattle, horrible work environments, people, lots of slaughtering, lots of blood. They used to put ice water on the ground to try and minimize the risk. So people would end up having, their feet would sometimes need to be amputated after a while. So it was a really horrible circumstance they were working in. Many of you may be familiar with Upton Sinclair's novel, The Jungle. There was a whole bunch of journalists at that time called the muckrakers, and he was one of them. And he wrote the book, The Jungle, about the Chicago meat trade. It was horrible. It was disgusting. Most people who have read it hate it because it just affects them very profoundly. Not because it's a bad book, but because it is so disgusting in its details. Teddy Roosevelt read it too. And I'm simplifying maybe a bit the history here. It wasn't as simple as he read the book and he came up with the decision. There was a lot of lobbying going on, a lot of strong physicians behind the push. But this was one of the pivotal areas in which caused him to create the Food, Drug, and at that time Insecticide Administration, later becoming the FDA. So this was the genesis of the FDA, and it evolved a lot over the next 20, 30 years. But this is how we have had the FDA here. A lot of other things happened along the way since that time. We got placebo control. So we talked about Dr. James Lind doing a controlled trial where he had people in different groups and tested one treatment versus the other. A placebo control is where one of them has a placebo, which is no drug. And of course, you want to not be in that group if you can avoid it. You can go further and you can randomize, which is the next step here. So randomization is where you say, we will randomly assign a patient who's on a trial to one group or another group. This sounds like a small deal today. We're all very familiar with randomized trials, but it was a really big deal when it first happened. Nobody had thought about reducing the bias by randomizing the patients. The next step was treatment blinding. Okay, so now you've got placebo, you've got randomization, but the physicians knew who was getting what. And if you have a vested interest, one way or the other, in the outcome, then the blinding gets rid of that type of error. These are all statistical errors. We can go into the discussion of what type of errors are related to which of these elements, but each of these reduce type 1 and type 2 statistical errors, leading to better and better clinical trials. So these all happened in the 1920s, 1930s, 1940s. A lot of very smart physicians played a big role. The FDA played a big role in getting us to the place where we today have the modern placebo-controlled, randomized, blinded clinical trial. I think it's good here to pause and talk about thalidomide. I'm sure many of you have heard about thalidomide, know the story, but it really is a big success story in many ways for the FDA. It was patented in 1954. In Europe, it was approved in 1957. It is an antiemetic, so it prevents vomiting. So a lot of women wanted to use it for morning sickness. Dr. Kelsey, Frances Kelsey, did her Ph.D. First of all, it was unusual at that time for women to get Ph.D.s. She got her Ph.D. because her name sounded like a boy's name, not a girl's name. So she got there, and her advisor said, "You're not a guy." I mean, literally, that was the opening meeting that she had with her advisor. She had worked on transfer of chemicals through the placenta in her Ph.D., so she was very familiar with the fact that things could go through the placenta. At that time, the common belief was that the placenta was impermeable. So everybody knew thalidomide had potential risk, but because they believed that the placenta was impermeable, they said it was okay to give it. We all know the story of what happened in Europe. Tens of thousands, hundreds of thousands of kids with birth defects. She was the one who said no in the US. And there's a lot of great stories about her online. I highly recommend you read it. There's a book about her as well, and it's very worth reading. It's a really terrific story. She was the one who refused to do it. She held on, she delayed it, and by the time the results came back, we knew it was a problem. And the FDA really stood out as a really big success story for what they were able to achieve with thalidomide. I'm going to fast forward. So I tried to take you a little tour of the past and give you an idea of the history of how clinical trials came from biblical times to the post-World War II. You know, since then, I'm going to fast forward. There's lots of innovations taking place and really talk about what's happened in the last 15 to 20 years. And I'll give a little description here and talk through what does it take to get a drug to market today. You know, clinical trials take a long, long time. Some of the drugs that we're talking about now coming on the market were first discovered in academic labs 15, 20 years ago. After a few years, they seem promising. They make it to a pharmaceutical company or a biotech company or some commercial entity, maybe 10,000 of them, and they analyze them. Nowadays they use high throughput screening with gene chips and other ways. In the past, it used to be an extremely laborious method, but a few years later they get to preclinical, and I'll describe that in my following slide in a little more detail, what preclinical means. Out of the 250, only 5 make it to human clinical trials. And of that, only 1. So 1 out of 10,000 compounds, 15 years later, makes it to an approved product. And even then, probably 1 out of 3 of those really goes to be very successful and make a big difference. So this is a very hard problem to solve. The work starts at the preclinical. I mean, you've got 10,000 compounds, you've identified 250. Now you just need to optimize. Okay, what doses? How does it have to look? What's the chemistry going to look like? How am I going to? What concentrations seem to have an effect on a Petri dish? You're not even at an animal model yet. You then have to think about it in an animal model. How good is the absorption? How's the distribution in the body? Is it all going to the liver? Is it actually going to where the drug needs to get to? How does the metabolism of the drug take place? And how is it excreted finally? So all of those need to be really carefully considered because they also give you clues to how effective it's likely to be, how toxic it's likely to be, how the interaction is likely to be with other compounds. All of these need to be considered. You then have to say, what am I going to mix it with to get it where it needs to go? Do I need to put it with another chemical or a biochemical that then attaches to a particular protein, that then attaches to a particular location or a target, which then can deliver a problem? Many drugs die between step 2 and step 3 because they just can't be delivered. You then, at that point, you have to then go and do pharmacokinetics, pharmacodynamics. Okay, if I keep increasing the dose, how much of it's in the blood, how much of it is effective. You check that, you study that very carefully. You do many of these steps concurrently. You think of the formulation that's going to be administered, and then you finally think of what the toxicity is going to be, and then you think about how am I going to make it. So, you know, at this point, 4 or 5 years have gone by. From the point, probably 7 years from the point of which it was discovered in the academic clinic. This is already 7 years later. So when we talk about the genomic revolution, and sometimes some of us wonder, why isn't it that there isn't an avalanche of drugs that have come about from the genomic revolution that we've been talking about for 20 years? It's because of this. I hope in the next 5 years we see a lot more come out from the work that's happened in the last 15. That said, there's been a lot of spectacular successes that have happened. In oncology, we know, I mean, non-Hodgkin's lymphoma has been a terrific success story in oncology, as well as many others. Diabetes, insulin, single-handedly transformed diabetics' lives. I mean, we know in other areas, arthritis, osteoporosis, the silent killer disease in women, there's a lot of solutions for. The pacemaker in cardiology, big difference before and after. The catheter, a very simple concept, but fundamentally revolutionary. All of these have had clinical trials to get them where they are today.